Hi, this is Duncan Ferguson. In this unit, we're going to talk about the cellular mechanisms associated with the mechanism of action of steroid hormones. Specifically, we're going to focus on glucocorticoids and mineralocorticoids, but many of the things generalize to the sex steroids that you'll hear about in separate units. So the cellular mechanism of action is basically can be broken down into two types. Those that are nuclear in nature are called genomic, and those that are non-genomic. And so the classical mechanism of action for steroids is generally considered to be of the nuclear variety, and this generally, in our case, means that they're evolving a longer time course, usually hours to days. We'll go through that time course a little bit later in a few slides ahead. But we were talking about glucocorticoids and mineralocorticoids, receptors mediating these more specific effects for these two steroid hormones. Well, sort of jumping ahead, we know that some of the glucocorticoids have high affinity, like cortisol has a high affinity for the mineralocorticoid receptor. So you might say, well, how on earth can that be if you want a specific action? And so what the body's figured out here is that in tissues that are targets of mineralocorticoids, there are, and these include the kidney, the intestine, and the salivary gland. Cortisol is metabolized to a less potent, it's not impotent, but a less potent cortisone by an enzyme called 11-beta-hydroxysteroid dehydrogenase. And this allows the mineralocorticoid effect, and generally they're circulating in lower concentrations than glucocorticoids, to predominate. Now the non-genomic effects are, from a relative standpoint, relatively new. We've suspected they've been there for some time because they have a more rapid time course. We know have used glucocorticoids in the context of treatment of shock, for example. But these are via plasma membrane receptors or other organelles, and we'll show you a graphic in a couple of slides. But these do not require activation of the nuclear receptors. In this slide, we're going to basically take a look at the big picture, if you will, with regards to nuclear receptors that affect a lot, that is the mechanism by which many different ligands or binding proteins or binding compounds or hormones have their effect within the body. And so you can see on the left the steroid receptors, which includes all of the glucocorticoid, mineralocorticoid, progestins, androgens, and estrogens, that is the sex steroids. These tend to be, or always are, homodimeric, meaning that it takes a binding to a receptor, and we'll go over the top structure up here in the next slide in more detail with regards to glucocorticoids, but it takes two receptors of the same type to interact with the DNA binding sites. Whereas in heterodimeric systems, for instance, thyroid hormone, which we'll talk about in a separate unit, they're always heterodimeric because it's the ligand or the whatever the hormone is binding, and then there's the retinoic acid receptor. So it's two different types of receptors, and they influence each other's action in different ways. The important thing to recognize is that's what heterodimeric means, and we'll talk about more detail about the glucocorticoid receptors next. Okay, so let me walk you through the process of a steroid hormone up here at the top left binding to its receptor, as we call that. That hormone will come through the membrane, or it's transported through the membrane, and we're assuming that's happening, and then it's going to interact with the receptor, in this case in the cytosol. That's called the ligand binding domain right here. And then there's a section of that peptide that will also be capable of binding to DNA. We call, obviously, that's the DNA binding domain. And now turning to the DNA itself, down here, you can see that there is a section we call the hormone response element, or in this case it's the steroid response element, or if it was thyroid hormone, it would be the thyroid response element. That is the part that recognizes the DNA binding domain. So that goes there, like that. And the target gene is any gene that has this hormone response element and is therefore going to be influenced by the hormone. So then if you look down to the bottom, you can see that here we have two glucocorticoid, let's say, hormones, but it can be any steroid hormone. It has to be the same receptor. Whether it's glucocorticoids, it's got to be two glucocorticoid receptors or two mineralocorticoid receptors, and they are dimerize and bind to the hormone response element, and that leads, of course, to gene transcription, which is messenger RNA, and then eventually protein synthesis. So let's repeat that in textual format here. Steroid hormone basically diffuses or it's transported across the cell membrane, enters the target cell. The hormone binds to its receptor, whichever one is it interacts with normally. And then that hormone receptor, that's a non-covalently bound complex that then leads to activation of that receptor. That activated hormone receptor complex enters the nucleus at that point. It's been in the cytosol before that point. This complex then finds another one, homodimerizes, and then binds to specific DNA sequence we call the hormone response element. That becomes a transcription factor, meaning that it regulates messenger RNA transcription. And then that means the new mRNA is produced, leaves the nucleus, and is translated to a new protein, and that new protein has some biological action that we would call a steroid endpoint, if you will, a steroid hormone endpoint. For example, we'll talk about lipocortin as a protein that inhibits phospholipase A2, and by doing so, it has one of its effects as an anti-inflammatory hormone. Just so you have adequate background on how the nuclear receptors in general are regulated and some of the terms that are used, I think it's well worth taking a step back at this point and talking about them. We talk about upregulation. This is actually any transcription rate to messenger RNA above the basal rate. That would be upregulation. This should be distinguished from what you might hear in other places called receptor protein upregulation or downregulation, which means that that protein is made in more or less a quantity as a secondary response to, say, stimulation of the receptor. Downregulation, of course, is the movement below the basal rate. This needs to be distinguished from the fact you have some genes that, when bound by the let's say hormone, are downregulated, and therefore they would be downregulated under conditions that are normally expected to increase the rate of transcription. Those are negatively regulated genes. So that means the effect of that hormone, for example, is to reduce that activity of that gene. Whereas you can also have impairment of activation, which means the prevention of or the reduction in activation of transcription by other factors. And this is sort of means that there's all kinds of ways, sort of networks, that can impact the final effect, which is to produce protein. And then repression is the term that's used to regarding the reduction in the rate of transcription to a level below basal, and that's usually because it recruits repressive factors or complexes. And this actually is a pretty important mechanism of action you'll see with regards to thyroid hormones, and we'll talk about that in a separate unit. Okay, in this slide, which is also a little more complex, let me walk you through it, reiterating some of the things we've already said. So we have our glucocorticoid, which we call GC, so that's a cortisol. And it finds its way through the plasma membrane by either some specific transport mechanism, or it can just do it by diffusion. And then interacts with glucocorticoid receptor in the cytosol. And then that can be moved into the nucleus, and as we said, interacts with the DNA in the way we talked about in more detail down here. So we're in the nucleus, it's interacting, it can have activating effects, as this over here is called transactivation or transrepression. It's influencing the effect of, or the amount of a certain gene being made, messenger RNA, then protein, upwards or downwards, and depending on how things are programmed. You can imagine that these would be, this is the important point, this mechanism has a slow to medium slow sort of time frame. Let's say hours to days. This contrasts with the right-hand side of the slide where a glucocorticoid comes through the membrane, interacts with the membrane receptor, membrane glucocorticoid receptor, and then has specific effects. And these include some that could be altering membrane currents, so it has electrical effects, electrically excitable membrane effects, can alter phosphorylation, or can alter calcium. And the key point is that these are not involving DNA or interaction with DNA or messenger RNA synthesis, and therefore these can be relatively rapid. These are the types of effects that we think occur when we give really high doses of glucocorticoids in shock. So on this slide, let me give you a specific example. We talked about it a little bit in a couple of slides before, but so we have our plasma membrane. We talked about that as one mechanism, but I'm going to talk, so that's here, and that can be the shock effect. Let's not focus on that right now. Let's talk about the binding to the cytosolic receptors, and then interaction with the nucleus, transcription, and then take a specific protein, in this case lipocortin. Lipocortin is a protein that has an inhibitory effect on an enzyme, and I believe you've learned about this enzyme, phospholipase A2, which is capable of making or releasing arachidonic acid, and arachidonic acid then feeds into enzyme pathways that produce prostaglandins, leukotrienes, and platelet aggregating factor. Lipid mediators of inflammation. And so this mechanism is one example of a protein and how its synthesis can be affected. In this case, activity, it's increased, the lipocortin is increased, but it has a negative activity on an enzyme which decreases the lipid mediators of inflammation. So in summary, the steroid cellular mechanisms, we have nuclear or genomic, and we have non-genomic. And the non-genomic ones basically are usually through plasma membrane receptors that are specific for glucocorticoids. Nuclear glucocorticoid and mineralocorticoid receptors act as homodimers, meaning it takes two to tango, two to bind at the hormone response element on the DNA. This activation can lead to an increase or a decrease in synthesis of specific proteins. That's how we see the effect. And then the non-genomic mechanisms are generally more rapid than genomic mechanisms with regards to their time frame.